Hello everyone, this is Praveen from the Department of Chemical Engineering at Auburn University going to present on my topic co-transport of ethanol and carboxylate ions in PEGDA-based cation exchange membranes. As we all know, the global carbon dioxide emissions is on the rise from the past 200 years, and this has also resulted in the increase in global average temperatures. To mitigate this negative effect, we need to look for ways to tap and utilize CO2 into various useful chemicals, and one such proposed solution is to use CO2 reduction cell, where that absorbs sunlight and reduces CO2 into valuable chemicals. But unfortunately, the membrane suffers from the lack of permeability of necessary ions, and while also prevent the permeability of certain CO2 reduced products, which can hamper the efficiency of this device. And this deficiency is properly illustrated uh, by this radar diagram shown here, where the CEMs is the abbreviation for cation exchange membranes. Previously, people have studied the copermeation behavior of methanol and acetate to Nafion based commercially available cation exchange membranes. So now we are planning to prepare model cation exchange membranes, uh, which is PEGDA based, where PEGDA is acting as a cross linker, while there are other co-monomers such as sulfonated and uncharged co-monomers, which can effectively tune the properties of this membrane. And to measure the permeability of this membrane to formate ions, we use uh, uh, we use custom built diffusion cells, which are shown below. This diffusion cells are named as feed and receiver cell, and the membrane is kept in between these cells, and the feed cell consists of potassium formate solution, and the receiver cell consists of pure water initially, and the receiver cell is clamped with a conductivity meter probe to measure the conductivity. So as time passes, the formate ions diffuses from the feed to the receiver cell, and the conductivity meter probe measures the concentration of formate ions which have diffused to the receiver cell, uh, uh, to the cation based membranes, and the permeability of these formate ions to the membrane is measured through a Suda's model equation, which is shown in the right. And so far we have prepared two types of membranes with varied sulfonated co-monomer content. One is with 0% co-monomer content, which is referred to as PEGDA 100 or 100 mole percent PEGDA, uh, where in the graph we can see the permeability is almost constant for increasing concentration of formate ions to PEGDA 100 membrane. Um, while the other membrane which we have prepared is 32 mole percent of sulfonated co-monomer and 68 mole percent of PEGDA, and here we can observe the permeability of formate ions gradually increased with increasing uh, increasing concentration of formate ions. So in future we are planning to prepare other membranes which also contain uncharged co-monomers such as phenyl ether acrylates, which are shown here and which are abbreviated as EGPMA, EGPEA and PEGPEA, which can efficiently tune the fractional free volume and other desirable properties for this membrane. And we will also measure the transport properties of these membranes uh, with formate and ethanol to these membranes, and also perform desired membrane characterizations to measure the properties of these membranes. At last, I would like to thank the funding source DOE and my research group and Auburn University for the support given to me for the project. Thank you so much.